Hello, thanks for joining us this morning, and thanks to VitiCulture for hosting this webinar and also hosting the VitiCulture event online. It's a great show last year down at Plumpton, and it's excellent to be able to continue the show this year in this virtual format. So my name's Ant. I'm a technical development specialist at Fargro, and I'm joined today by Damien from Agrauxine. So today we'll be looking at Romeo, which is a new biofungicide, specifically for this presentation within viticulture. Romeo is a product that we released onto the UK market about 1.5 years ago, but it was only at the start of this year that we got a label extension in the viticulture. So we'll be looking specifically at how you guys can integrate it into your programs. So for those of you that haven't heard about Fargro, we're a horticultural distributor based just outside Arundel, and we've been supporting horticulture for 75 years in all forms. So that's all forms across quite a diverse array of crops and across quite a diverse array of products as well. So we do everything from sort of added value and sundries all the way to production equipment and asset finance. So we're really trying to be a sort of one-stop shop for horticulture. The key area that I'm involved in is our technical department, where we work with our plant protection products and IPM programs and have quite a focus on biopesticides. So within the technical department, we have all sorts of people from PhD entomologists right down to more sort of practical ex-growers that are all out in the field, supporting growers with their IPM programs and helping them get the best from our biopesticides. So it's quite a wide range of services and products we offer. So please do, after you've had a chance to have a look at all the talks, come over to our virtual stand. There'll be one of us on the stand all day from Fargro, be it an ASM or a technical member of the team. And hopefully intermittently throughout the day, we'll have some members of the team from Agrauxine on the stand. So the tech department itself have quite extensive experience in IPM. So we were one of the first companies to start really working with macrobiologicals in the UK in the 1990s, and the first company to bring a biopesticide to the UK market in 2009 in Serenade. So alongside that, we currently have one of the widest portfolios of biopesticides in the UK. So we have quite a lot of experience and also quite a wide breadth of knowledge in these products. And so we use that experience and breadth of knowledge to help us form our IPM programs and inform our recommendations when we make them. So when we're recommending products and implementing programs, we're always doing it from that point of view of following the IPM paradigm of having a really strong base on which to start your program. So good hygiene, good varietal choices, constant monitoring, looking for pests, recording, using traps, and ensuring that the environmental conditions are never going to be overly conducive for pests and disease. We work very closely with technology providers like 30 megahertz to use agri-tech and cloud-based systems to help our growers with their decision making on what products they use. So we use NZ to create a decision support system on when to apply certain biopesticides. And then we move into the realm of biopesticides and chemical control, which we obviously have higher up the IPM pyramid. So Romeo is a specific product we're going to look at in this presentation, but in terms of products that we have available to the viticulture market in the UK at the moment, we've got stuff like Lepinox, which is a Bacillus thuringiensis, Tegro, which is Bacillus amyloliquefactions, and a product called Blockade, which is a wound paint. A lot of these products are what we call national exclusive products, so they'll be available right up and down the UK. We distribute through all the major agri distributors as well as the horti distributors, so you can get these through Agri, Hutchinson's and Agravista. So hopefully wherever you are in the UK, you should be able to get hold of these. So it's a really exciting time in UK horticulture, and we especially see that viticulture itself is one of those key exciting growth areas. I grew up in Eastbourne, so it seemed like every summer when I was growing up there was a new planting of vines going up in the fields. So it's really important that we see that growth industry in viticulture as well as the growth industry in biopesticides and bring them together so we can continue producing the highest quality UK grapes with sustainable crop protection methods. So that was a brief intro to Fargro, and like I said, please do come and visit us on the stand for a bit more info on the products and to get to know us a bit more. So I'll hand over to Damien now for a intro to himself and Agrauxine, and then we'll look at Romeo in a bit more detail.
Yeah, thank you, Ant. So I don't have especially a presentation to go through on Agrauxine, but I will just introduce you a little bit to who I am and to the company. So I'm Damien Verger. So I'm a development biocontrol manager internationally for Agrauxine. So I arrived in Agrauxine about a year and a half ago, and before I used to work at Bayer's, working in field as a technical help for field trials. So regarding Agrauxine, so Agrauxine is the business unit of Le Safre, and Agrauxine is dedicated to plant care. So Le Safre is known to be one of the world leader on yeast. So it's recognized as an expert regarding fermentation process. And so Agrauxine relied on this fermentation process expertise to develop and manufacture products, so from microorganisms for bioprotection. So we have a range of biocontrol products, and also for biostimulation, as we also do some biostimulants. And so yeah, as Agrauxine, our ambition is to contribute to the establishment of a sustainable and performing agriculture. The philosophy of the company is really to only to do some products that can be used for organic farming, and all of our range of products is based on this philosophy. So that's it for me.
Okay, that's great. Thanks for that, Damien. So we'll move into the bulk of the presentation now. So yeah, Romeo itself is a really unique and interesting product. So can you explain to us a bit about sort of background research that went into finding the right strain and understanding their production methods and the sort of fundamental science behind Romeo and cerevisane itself? I don't know if you've got some slides to share at this point as well.
Yeah, I will show you the slides. Here it is.
Great.
I don't know if you can, if what you're seeing here, but...
Yep, we've got the slides there.
Okay, perfect. So first I'll start with the basics on what is yeast, as it's not especially known by everyone here, but just a quick reminder. So yeast is an ovoid and spherical unicellular microscopic fungus. So very small fungus. The most well-known species is Saccharomyces cerevisiae. And there are many different yeast species and even more different strains. As Le Safre, as an expert in yeast production, they have a collection of more than 6,000 strains, which gives quite a good example of the diversity of yeast strains that you can find. So regarding Romeo, and why did we start to think about using yeast for biocontrol? So in the 70s, there were the first scientific publication showing the effects of glucans as elicitors. So it was really research work. They showed the effect of showing some defense mechanism activation, first on animals and on plants as well. And then there was some kind of a scientific fad around the subject in the following decades with many publications. But first it was only in scientific world before it went to the industrial world, and to then be to think of other uses. So here, as an example of a 70s article that worked on glucans and difference response in pathogen hosts. And so afterwards, it went to the industry and to Le Safre. The R&D team of Le Safre heard, of course, about this fad around the glucans, and they knew that the cell walls of yeasts were rich in these glucans, and so they're starting to trying to select some specific strain that were rich in these compounds to then use them for what will be then Romeo. And so, what is cerevisane? That is the active ingredient of Romeo. So cerevisane is part of cell wall of Saccharomyces cerevisiae, so a specific strain that was selected by Le Safre because of its capacity of having a lot of these compounds in its cell walls. So it's a natural product as it's based on yeast, a selected strain, and a non-living product, as it's only the cell wall of the yeast. It's not a living yeast. It's a separation that is done. I will talk a little bit about it afterwards. A separation that is done of the yeast, of the inside and the outside of the yeast. And so in 2006, the Le Safre R&D pipeline, they patented cerevisane, and now we have a very good control fabrication process of this specific ingredient based on Le Safre expertise on yeast. So just to explain you a little how is Romeo processed. So the base is a unique strain, so of Saccharomyces cerevisiae, named LAS 117. So they will do multiplication as it's always been done in big fermenters like these ones. And a specific part that is a unique knowledge of Le Safre is then the extraction and the filtration, separating the yeast internal part and external part. And even more than just separate it, it's also to keep this specific structure of the cell wall so it can contain the 3D shape of the yeast, and so that it will maintain the integrity of the cell wall, and so the plant will recognize more this cell wall. And this is what is cerevisane. That is the active ingredient of Romeo. So a very stable formulation with no conservation constraints and very easy to use. As here, a little example, it mixes very well with water, and it's a WP formulation. So here a summary of cerevisane. So it's a cell wall, so it's not living. It was approved by the EU as a low-risk substance. So all the products from Agrauxine. It's now registered in many European countries and also in the US. There is no MRLs, which is also a very important point, and it's of course organic farming compatible. So it's mainly composed of cerevisane, almost only cerevisane. The rest is actually some water remaining, as it comes from a living organism. And so the formulation is WP. You have a 2-year shelf life at room temperature, so as any other chemical. it's very stabilized the way it is in the end as it is dried. We'll go further on the use, but it's used preventively on foliar applications at low dose rates. There is no phytotoxicity, of course, and as it's a non-living product, it's very easy to use it in tech mix with chemicals. It will not affect the biology of the effect, as it's a non-living product. And regarding the packaging, it looks like here on the top right, so 1 kilogram bottles and 8 pots per card box.
Great. So yeah, if I jump in here, Damien, thanks for that little introduction. So I guess two of the key points that I take from that, as someone that works with biopesticides a lot, is the storage and tanks mix compatibility of the products really good. See, a lot of the biopesticides we work with tend to be entomopathogenic funguses, so they don't really tank mix, and their storage requirements have to be quite stringent. So it's really good to see a biopesticide coming through that's less stringent on those categories. So in terms of its mode of action, systemically acquired resistance and induced systemic resistance are talked about quite a lot and becoming more and more common over here in the UK. So I assume we're going to go into this, but can you just talk us through a bit how Romeo specifically engages certain genes and what actual physiological processes that then undertakes within the plant and how that protects against diseases?
Yeah. As I said before, first researcher worked on these specific glucans, and so glucans, mannans, chitin, all these compounds that are part of the cell yeast wall, they will be received from the plant as a mimic of a microbial attack. So it will be percepted by the plant that has some specific receptors. And so then it will activate a cell signaling cascade that will then induce some defense genes that are well known now, called as PR proteins, and some phenolic compounds as well will be produced. So it induces many different genes. One of the study that was done here by the University of Bari in Italy is the expression level study of PR1 and PR4. So PR1 and PR4 are some marker genes of the defense pathways dependent on salicylic acid and jasmonic acid and ethylene. These three molecules are very well known in the research in the pathogen as pathogen response from the plant, molecules that are produced when they are attacked by different types of pathogens. And so here, one day after treatment, Romeo on foliar applications on Grapevine, there is an expression level shown here regarding the PR1 and PR4, showing that Romeo's induced the expression of these specific gene, and so of the pathways of pathogens. So you have here PR1 and PR4. These specific molecules will and metabolic changes will activate some defense in the plant and will be effective in the defenses of the plant. There are many different pathways of the plant that the plant is using all its capacities to defend itself against the pathogens. One of the pathways is to produce phytoalexins. We showed as well that Romeo is able to activate this stimulation of phytoalexins, as these molecules will be kind of toxic against the pathogens. So here you have colored in blue or white blue on cells. These are the phytoalexines activated by Romeo 3 days and 7 days after treatment, showing also that there is a production of this phytoalexine from 7 to 10 days after the treatment, which is an important point that we'll go through after. So other pathways as callose production. What is important to remember is that Romeo and cerevisane will be detected by the plant. All the cells will detect, all the foliar cells will detect the active substance, and so will induce some genes that are very known to fight against the diseases. And Grapevine is very receptive to this to these types of signals, and so then the plant will be protected against further pathogen attack, and that's why it needs to be preventive, as when you treat before the plant gets attacked, the plant will produce all the different mechanisms to defend itself, and then when the pathogen will arrive, it will not be able to do its cycle as good as it was if there were no induced defense before that. So here you have an example of callose deposition visible. So it's another way of the plant to avoid the pathogens to enter in the cell. So here it shows an internal colonization of Plasmopara viticola, so downy mildew, 7 days after inoculation. So on the two top pictures, without Romeo treated before, and on the bottom pictures when treated with Romeo, and all the blue dots that you can see on the bottom left and on the bottom right show with an arrow are callose deposits that are mainly deposits placed by the plant around the stomatas, as the stomatas are entry doors for the downy mildew to go inside of the leaf and then perform its infection cycle. So here are some nice pictures of internal colonizations of downy mildew after inoculations of Romeo and without, with preventive foliar spray, of course. And here are some other techniques to show the internal colonization of Eryspinacator, so powdery mildew, inside of the leaves, colored in blue. So the hyphae you can see on the left when untreated, and on the right when treated with Romeo, as all these defense mechanisms are used by the plant to fight against different pathogens. So downy mildew, powdery mildew as well, and botrytis as well. So a quick summary of the mode of action. Against its yeast origin, so it's based on cerevisane. It's not living, so there is no direct effect on the pathogen. The idea here is really to use the whole potential of the plant to fight against these diseases. So it acts as an elicitor. It will stimulate the salicylic acid, jasminic acid, and ethylene pathways. It needs to be preventive. So before the first symptoms, but also before the first contamination. So you will have not to wait for the first symptoms to arrive to treat. It's really, the idea is to treat before that, as we know that the first symptoms will appear only for a few days after the first contamination. A very important point, as I said, is that it has a broad spectrum against fungal diseases. and there is an efficient protection 24 hours after the treatment during 7 to 10 days.
Great, thanks for that, Damien. It's really interesting to see and understand a bit more about systemically acquired resistance and see how that upregulation in certain genes is linked to certain metabolic changes that happen within the plant, and then link it to how that's actually influencing the ability of a pathogen to infect. So thanks for that. Just one point that you mentioned in there was on how the foliar cells that perceive cerevisane and then upregulate certain genes. Is there any sort of intracellular communication within a leaf, or is it very important that you get good coverage with the product to hit as many cells as possible?
Yeah, very good question. It's a question that is often raised by the people that I can understand. The idea here is to have a very good foliage cover, as all the cells in their own will participate in these defense mechanisms. There is some signaling pathways, but they are not as efficient as if you're asking directly the cell that is next to the one that is activated to be activated. And so that's why, from what we saw, and as most of the products, a good foliar application and a good spraying quality is very important to then have a good efficacy of the product.
Great, thanks for that. Yeah, so a key barrier with a lot of biopesticides is that ability to tank mix, and one of the key strengths here with Romeo definitely is that ability to integrate it into a program, not just perhaps within tank mix, but also within alternations with conventional chemicals. So with that in mind, should we go through a couple of trials and look at some data on some key foliar diseases in viticulture?
Yeah, of course. So just regarding the compatibility, so as it's a non-living product, we have really no issue regarding mixing it with other products, as it's a very stable product and as it's a non-living product. So we did many different tests to compare it, the compatibility. And so if there is any raised question regarding a product, if it's compatible or not, we have the data on this, or we can do some tests. So as I said just before, an important thing with Romeo is that it has this capacity of targeting different diseases as powdery mildew, downy mildew, and botrytis. So we did many different trials, GEP trials, as it was needed to register it as any other chemical product, because it's important to notice that it's Romeo was registered as a normal chemical product, even though it's a biocontrol one. So here you have an average of 8 GEP trials done between 2009 and 2015. So assessments of severity on leaves and on bunches at the beginning of the infection. So with an average of 10% of severity on different locations. So in Europe, in France, Italy, in Spain, on different cultivars, and with an interval of application of 10 days average. That's our recommendation. And what can be noticed here as well is the water volume mixture that is very different from around 100 liters per hectare to 1,000. As in countries, this specific water volume mixture habit is very different. So we didn't see any issue regarding this water volume mixture. So here in this average of 8 trials, the idea was just for you to understand the intrinsic efficacy of Romeo as it's the product on its own compared to a reference program. So here the reference could be mancozeb or copper or methylene regarding the different countries and trials. So a good intrinsic efficacy at the beginning of the attack, As you can see here, it represents the efficacy on the leaves and the bunches, so up to 70% of the leaves and 80% on the bunches. So it will not be sufficient alone during all season long, as we are only working on the capacity of the plant, and the capacity of the plant will be limited at a certain point. But it's really interesting how much we can show this specific efficacy on leaves and bunches on its own, only treating with Romeo, reaching very good efficacy. So it's a new solution that needs to be integrated in the global program to optimize the current program performance. And that's what was done already in some trials, as here in Hungary, in down mildew. So with the conventional reference, so with Polyram, Orvego, I don't know if all these products will ring a bell for you, but classical Methyram, Dimetomorph, or Ametoxetradine products. So basically reference conventional references, and then reduced conventional references with reduced doses of Dylan at the end, and of Polyram at the beginning, and then these same reduced references added with Romeo at the beginning and at the end of the cycle. Then compared it with the full dose rate of copper. Then copper plus Romeo to optimize the copper efficacy, and then Romeo on its own to have a good full trial. So here are the results that we can achieve. So on the left you have the percentage severity on the leaves. In gray on the untreated control, the evolution. It reaches about 44% of severity on the leaves. When you have Romeo on its own, you already have some statistical differences, as it's reduced to 31%. Then you have the copper in blue, that leads to 20% severity on leaves. And then you have the programs. So you can already see here that the copper not reduced plus the Romeo added will significantly reduce the percentage severity on leaves, as well as the reduced reference program and the reduced reference program plus Romeo. So, and you can see the same tendencies on the percentage severity on benches on the right. So quite a high pressure on leaves and on benches, and a very good efficacy of the program when you mix Romeo with copper compared to copper only. And of course the conventional reference works very well as well, and we reached with copper plus Romeo an efficacy that is equivalent to the conventional reference. So here the same idea of the intrinsic efficacy on powdery mildew. So here it's an average of 28 GP trials for about 10 years of trials. Same thing on leaves and bunches, the percentage of efficacy. So about 76% efficacy on leaves and 68% on bunches. Here the references compared were of quinoxyphene and a lot of sulfur as well, as it is mainly used now. So a good intrinsic efficacy also, but still a sufficient alone during all the season. And again, a very interesting solution to be integrated in the program. So that was done In here, you have an average of 10 GP field trials in France, Italy, Spain, and Portugal. Doing some strategies, mixing it or alternate with the sulfur and Romeo. So here you have, with the alternation on the left, the percentage of efficacy on severity on the leaves on the left and on the bunches. So we can see that we reach a similar efficacy to sulfur, alternating sulfur and Romeo compared to full sulfur. And when tank mixed, you have a 5% increased efficacy in comparison to the sulfur, reduced at 15%. So here you have in blue the reference sulfur dose. In blue and white, the sulfur reduced at 50%, and when mixed with Romeo, you will see the adding effect of cerevisane. Here another example of a tank mix program. Here not with sulfur, but with classical chemicals. So here you have the untreated control in black. You can see that you will reach by the end of July a percentage of severity on leaf of 50%. And then it's compared to the reference program. So you have all the different molecules that are used in this program at the bottom. So tebuconazole, fluopiram, metraphenone. Quite a very complete program. That works quite well here in dark blue. When reduced at 50%, you can see here that there is not so much efficacy as the full reference program. And when you add to this half reduced reference program some Romeo, it will lead to an efficacy that is equal as the full reference program. So a very interesting also dose reduction of this chemical program. You have here some picture of on the left some wettable sulfur program at 4 kilograms. So these pictures were taken in Italy last year actually. And on the right, this same program added with Romeo. And so you can see here that the preventive application will do a limitation of the sporulation, as you can see on the left, the sporulation starting on the powdery mildew, and on the right, not sporulated yet, and with a limited sporulation as well. Here, just at last, the field result for the Grapevine Botrytis trials, intrinsic efficacy. So here I have an average of 6 GP trials. Assessment of severity on bunches. So these trials were done in Europe as well with different cultivars, with different water volumes. Here the references were chemicals and also a Bacillus subtilis. So a medium efficacy alone, around 60% efficacy, as you can see on the top. So lower than chemicals, but on the bottom left, quite similar to the Bacillus subtilis reference. So again, a new solution to be integrated in program. So here is an example of a program that was done with fenpyrazamine. So in gray you have the incidence and the severity on the the incidence and the severity here. On the left, the incidence. So compared to a program with Scala in B and C, switch, and then fenpyrazamide in B and C, fenpyrazamide only in B, to be compared to fenpyrazamide plus Romeo and fenpyrazamide plus Bacillus subtilis. So here the data were collected just before harvest, and you can see that we have a very good efficacy of 1 or 2 application of Romeo after an application of the fenpyrazamide reference. As you can see here, the 10.5% incidence and the 0.5% severity. This program showed a very good difference compared to the green and white program of fenpyrazamide alone in B. So you can really see the effect on the C application of Romeo, even better actually than the Bacillus subtilis. So a significant additional efficacy of the Romeo application, and actually the best efficacy of this program compared to all the others, even the switch and the Scala, so the full chemical programs. An important point as well is to mention the impact on vinification, as here we are talking about reducing some doses or doing some programs of alternation. The reduction of sulfur has an impact on vinification. It was well shown already, and so here the idea was to see this difference in vinification with some full sulfur reference, with a full dosage of sulfur. You have on the last column on your right the total kilograms of sulfur. So 70 kilograms of sulfur. Then the modulated dose reduced by half. Then the even more reduced dose added with Romeo, and this same reduced dose without Romeo. So this was a trial done in Uni in France on Chardonnay. So regarding the efficacy, on the left, you have the evolution of the percentage of severity on the benches. So in green, blue, and red, it will show the evolution between the end of July and the end of August. So you can see that on the untreated control on the left, it goes quite fast in a month, from 25% severity on benches to 83% severity. Of course, the full reference sulfur program reaches very good efficacy, reducing almost the severity to 0%. Then the reduced by half shows still quite good efficacy. And then you have the sulfur reduced at 20 kilograms plus Romeo that showed very similar results by the end of August as the sulfur modulated dose. And you can really see here the added effect of Romeo regarding that on the right, you have the sulfur reduced at 20 kilograms that showed more severity on benches by the end of August compared to when added with Romeo. And so some tests were done afterwards on the vinification, so done by some different judges who tried to find the difference between the full program with sulfur and the Romeo plus reduced with sulfur. So there were 14 judges. So the sensorial analysis showed a slight improvement of wine organoleptic features with the Romeo strategy regarding the sulfur reduction, and they found it more fruity and less oxidized.
We've just got a couple of minutes left, Damien, if you'd just like to just sum up these slides.
Yeah, okay, no problem. So just another one that was done, vinification impact in Italy. So here they did kind of the same thing, comparing a full sulfur program compared to a sulfur program with some Romeo added. And so a significant improvement of efficacy. And what they saw afterwards in wine analysis and wine tests is that the sensorial analysis showed a slight improvement of wine organoleptic features with Romeo strategy, as the sulfur was reduced, as the last applications of sulfur were removed, and then it was added Romeo to add some more efficacy to the program. So again, another example on how you can improve the organoleptic features of the wine using less sulfur and choosing Romeo as a strategy in common with other products.
Excellent, thanks for that, Damien. I think it's really interesting to see sort of the full technical spectrum of a product and understand right from the beginning at the genetic level what's happening, and then see those metabolic changes, those physical changes within the plants, but then also see out of that lab work what effect that's actually having out in the field. So it's a really comprehensive sort of scientific overview of the product itself. So I'm very conscious that our time for the presentation slot is coming to an end. But as I said earlier, we'll be on the virtual stand all day. So please do come over to us and ask questions on the presentation or questions on the various other products we have coming through. Hopefully we'll have Agrauxine on stand for a bit during the day as well. So please do come over and leave questions for them. So thanks for tuning in this morning. I'll just hand over to Damien to do a final farewell.
Yeah, thanks for all. Thanks for inviting me, and it was very interesting. Hoping to have more exchanges like this. It was really good to maintain this event, and I'm really happy to be part of it.
Excellent. Yeah, well, thanks for listening, guys, and yeah, hop onto the stand and ask us some questions. Cheers. Bye.